Hi. Today let us get into the title Plant Kingdom Angiosperm, the last part of this chapter. Angiosperm means closed seeded plants formed in flowers. The flowers are the unique feature of angiosperms. And the second unique feature wherein you cannot see anywhere is double fertilization. And even the xylem vessels and phloem companion cells were not there in pteridophytes and gymnosperms, but there are few category like Neetopsida have got a vessels and companions. So special in the sense double fertilization and flowers for angiosperm. So basically angiosperms have got two major category, dicotyledons and monocotyledons. Dicotyledons in the sense they've got a two cotyledons. So if you take a seed, you can just split into two, that is dicots, wherein you cannot split like rice and wheat and all, you cannot split, you can just break but you cannot split. They are single cotyledonous, that is monocot. And when you talk about their root system, all dicots have got a taproot. So taproot means tapering towards the end. So you have just drawn here. Main primary root, secondary root and tertiary root hairs will be present. But here there is no tapering and all, a tuft of fibers. It is just like a ponytail kind of tuft, all or bunch. There is no differentiation of primary, secondary and tertiary, all a cluster of roots. And then stem with branches, mango, neem, banyan tree, and then ficus religiosa. These are all well branched, but in monocot they are all single stem. Basically, when you talk about monocot, mostly herbs and shrubs, but trees are very limited in number. But here the trees are more in dicots. Trees population is more. And when you talk about the leaf has got a dorsiventrally flattened. The dorsiventrally flattened leaf with reticulate venation. The veins are like entangled mesh. But here the leaves with parallel. See, it is isobilateral leaf. Like iso means same, bilateral, both the sides, or it will be standing like this. You could have seen in grass plant, like it will be like, and the veins are parallel venation. This is. And flowers, usually in dicot, flowers are either they have 4 or multiples of 4, tetramerous, or 5 and multiples of 5, pentamerous, both in sepals and petals. But when you come down to monocot, generally it is 3, that is 3 or multiples of 3, trimerous. And even this arrangement, the penta and tetra, wherein you have a sepal petal differentiation will be there in dicot. Here you just can't see the differentiation, both will be of same size, shape and color and all. So there it is called, there is no sepal and petal, and here it is called as a tepal. The unit is called a tepal, collectively it is called perianth. And the most striking feature and the difference between these two is, look at this floral diagram. This is the odd sepal. The odd sepal is posterior in position. Here the odd sepal is anterior in position. What is odd sepal? There will be a question. I'll just explain. Look at this. This is the mother axis. See, this is the place where, see, here this is the stem and here there is a flower. It comes like this. This angle is a mother axis. See, this is the mother axis. I have just drawn in both. This, imagine like this is the mother axis and the flower, this is the sepal which is facing this. This is the odd sepal in case of dicot. See, in case of monocot, it will be slightly twisted. It will be twisted like this. So this is the one facing the mother axis and this is the thing. Understood? Here this is the one odd sepal and here this is the one. So this is the very important feature. But in dicot family, Fabaceae has got a little twist and their odd sepal is anterior in position. That we will see under the families. These are the major differences. But common features like the angiosperm means, I said the very unique feature is double fertilization. I'll just give a gist of double fertilization. Fertilization between the gametes and the female gamete here. Here the pollen grain is a gametophyte which has got two generative nucleus and one tube nucleus. This generative nucleus are going to act like a gamete. So, once this is the ovary, the female part of a flower, ovary, style, stigma. Inside the ovary, there are some structure, ovule, and there is an embryo sac. Embryo sac has got 1, 2, 3 egg apparatus, and that the center one is a egg. And this is the polar nuclei clubbed to form a secondary nucleus, and this is antipodal cells. And here once it comes, it is released from the microsporophyll, and it just come and stays here in this area. And while it is coming, this is the tube nucleus, this is the pollen tube. The pollen tube was absent in gymnosperm, here it is present. It will be travelling via like this, and then coming, penetrating and getting into this via this small pore, micropyle area, this one. So, while coming, this tube nucleus will die. Only this two generative nucleus will be there. One generative nuclei will come and club with the egg nucleus. This is one fertilization. Another one generative nucleus is there now, that will just come and club with the secondary nucleus. So, the clubbing is 2 times. So, the one here, the first fertilization, and here they come and join the second fertilization. This is double fertilization. And after the fertilization phenomenon over, the side cells, synergids in egg apparatus, that will go off, and this antipodals will go off. Only this secondary nucleus, which has been clubbed with this generative nucleus with the 3N nucleus. Already 2 diploid will be there. Now the triploid condition will be alive. So now everything has gone. Only the fertilized egg and then this triploid. This is going to form an endosperm, and this is going to form an embryo. Understood? So this is the unique feature, the double fertilization. Nowhere in the plant kingdom you can see this. That is present in all the angiosperms, whether it is monocot or dicot, it is present there. Okay, and endosperm will be formed after fertilization. In gymnosperm, endosperm forms before fertilization. Here it forms after fertilization. So this is about the specific and uniqueness and importance of angiosperms. And one more small topic in this lesson is the cycles we discussed: haplontic, diplontic, haplodiplontic. I just wanted to give a gist of it. See here in angiosperm and gymnosperm, both the plant body is sporophyte, 2N. Only during the gamete formation they are undergoing meiosis and forming gametes, male and female gametes. So this is very short span in their life. They retain the haploid status. So the entire life cycle is with diploid. So that is diplontic cycle. And in case of pteridophytes and bryophytes, we have seen that half of their life cycle they have a haploid cycle, and half they will have a diploid phase of sporophyte and then gametophyte. So they alternate with each other, and that is haplodiplontic life cycle. And in case of a lowermost category like algae, even in fungi, there is a haplontic cycle, that is most of their lifespan with haploid, and only during the zygote formation they just go with the diploid. After that again they will go for a meiosis and then form a spores. So that is haplontic. Here in plant kingdom, the algae has got a haplontic life cycle. But in algae we have seen gradually it evolved from chlorophyceae to phaeophyceae and rhodophyceae, big the kelps and all, a big structure. They we have seen that the leaf like, and they're called fronds and all. So here there is some ectocarpus, scalps and polysiphonia and all exhibiting haplodiplontic cycle, and fucus exhibits diplontic cycle. So here this is very important in exam point of view. Some exceptional cases, you should be very thorough with, and odd sepal condition is very important. And what is the unit of perianth, and that kind of question will be again dealing under morphology. So this is about the entire angiosperm. With this chapter is coming to an end. If you have any doubt on this chapter on the whole, or anywhere in pteridophyte or angiosperm, please put it in the comment section. We'll just clarify things. And thank you very much, and please subscribe.